I think this is where we left off before Thanksgiving break. And so I've updated the slide set, by the way. I worked on it over break and I've updated it. So the current version in EduCAD is most updated from now. So this is the integrated plant health management idea, looking at the idea that plants certainly can influence which microbes they hang out with, and microbes certainly influence the behavior of plants as well. And so when you look at this idea, I thought this visual was pretty nice, just trying to impress on you that microbes are coexisting with plants in pretty much every part of the plant, except maybe the apical meristem region. You don't need to know these fancy words other than rhizosphere. I'd like you to know that one, which we've talked about already. That's the one on the lower right. So that's just the area of soil that is influenced by the roots. All these other terms are just trying to describe different parts of the plant body where we find microbes hanging out. And so now we're thinking about the endophyte idea if they're internal, but this word phytosphere means that they're on the surface, if you're wondering. And so certainly, just like with us, we have microbes that live on us. We have microbes that live in us, same with the plants. And they can be found everywhere, including the flowers and fruits. Okay, so that's the spermosphere idea of looking at flowers and fruits and pollen and all that. Okay, here is another diagram trying to get the gist across of how beneficial microbes can influence the plant and how the plant is interacting with them. So this acronym here is this abbreviation PGPR. That stands for plant growth promoting rhizobacteria. Rhizobacteria, it just means root related bacteria. That's what they mean by that term. It's just bacteria that hang out in the rhizosphere on the roots. You will also see similar acronyms like PGPM instead of R. PGPM is just M stands for microbes. So you'll see different versions of that acronym depending on which resource you're reading or who you're talking to. But this image is looking at several concepts, how you have the plant exuding certain nutrients that the microbes can use for their nutrition and energy needs. We've talked a little bit about quorum sensing already, how microbes can quorum sense with each other and cooperate to do some kind of group behavior, including making antibiotics that could kill or inhibit plant pathogens. You see also here that the plant is playing a role in quorum sensing. And so we talked a little bit about that before break as well, how the plant can certainly influence that whole process. So here's another example of a really kind of bizarre process in terms of the complexity here. Let me see if I can, it's not going to let me do that, is it? So what we have happening, if they have it numbered. So number one, you see the red kind of rod-shaped bacteria that's called Pseudomonas syringae. That's pathogen that loves tomato plant. It loves to attack tomato. So you can see what they're trying to show is that it's getting into the stomata. And so number two on that image is pointing to a stomata on the plant, and that the bacteria are able to gain entrance that way and can start to cause problems. So as this is happening, when they did this experiment, they, experimentally put Pseudomonas on the plant leaves and then watched to see what would happen in a tomato that was, growing in a soil environment. And what they saw was that the plant started to make this L-malic acid, number 3 here. So this is made by the plant. And it actually gets released by the root right after Pseudomonas attacks it. And then they're showing this bacteria, yellow. This little flower. They're showing this one producing black dots. These are chemical basically it serves a dual function. There are chemicals that will inhibit any certain microbes around the roof, but it also communicates back to the plant, hey, let me persist here, let me don't inhibit me. And so it's a way for the bee subtleist to it's a two-pronged thing. It communicates back to the plant saying, hey, I'm here, don't count me off, I'm actually going to do good things for you. And then but at the same time, bee subtleist is sending out chemicals, and they're trying to show here that some of the black dots, those chemicals, going to number 6 here, they're saying that there's other pathogens nearby that bee subtleist is helping fight for the plant. Then they're also showing that these chemicals will trigger, so the bee subtleist is producing chemicals that triggers the plant to close its stomata. So it's actually a little convoluted. The bacteria stimulate abscisic acid production by the plant. The plant ramps up abscisic acid, which causes the stomata to close. And so if the stomata goes down, that's preventing any more Pseudomonas from getting in. So it's just a way to shut the door on Pseudomonas, okay? And then they're also showing that here, they're showing another pathogen, number 8, and that what they're trying to show here now is that this tomato plant is successfully communicating to a nearby tomato plant, hey, I'm getting attacked, you're probably going to be next, you better ramp up. So, I mean, this is kind of complex and crazy, right? I mean, it's this interaction between first the pathogen and the plant, and then the plant and beneficial bacteria, and then plant to plant. And so very busy process here, all starting with the pathogen, the Pseudomonas in this case, gaining entry into the stomata, and that set off this whole cascade response with the plant. No, I think what they're trying to show there is inhibition. If you look at the, that's an alternator, or no, where's number 6? It staves off other potentially pathogenic bacterial strains. So they're saying that's another pathogen lurking. And so those black dots are the chemical being produced by the beneficial bacillus, and it's trying to kill off. So the little blurb up here explains what I just went through step by step. And so, and they also bring in the mycorrhizal fungi too. They're saying that it can be this communication from plant to plant, can be root to root, but often it's root, mycorrhizal fungus, root. And so you have fungus playing a role in getting the signal sent to nearby plants that they might be under attack next. Right. So really dynamic interactions going on that until fairly recently, we had no idea how complex this was between, the host plant, the associated microbes, both the bad microbes, the good microbes, and then the nearby plants that are also capable of. There's still some discussion on is the nearby plant, is it just kind of eavesdropping, or is there a direct purposeful communication between the plant under attack and its neighbor plants? That's still kind of up for debate and some interesting research going on. But you can definitely show that there is communication going on between the plants as well as between the plants and its microbes. So let me, I wanted to share with you, these white slides that I'm going to show you are actually part of a presentation that my postdoc advisor, Dr. Linda Kinkle, and her associate gave, and she gave me permission to use these in teaching. And so she's based at the University of Minnesota, where I did my postdoc in the Department of Plant Pathology. And so looking at these plant microbe interactions, one of the things that we're trying to do here is, if we can better understand the ecology of this, can we then manipulate the situation to benefit, create healthy plants, especially plants that are, high in nutrition and good for us to eat. And so then it's like, we want to be able to use these microbes as biocontrol agents. It's a better strategy than some of these nasty chemicals that can be used. And a lot of those chemicals, as we said, they're getting phased out. We can't use them anymore. So we're very interested in using the natural populations of microbes. Can we create like an army of biocontrol agents for the plant to help defend itself and stay disease free? So when you think about current agricultural practices here in America, by and large, the vast majority of our food is produced in a monoculture setting, right? Where you have just like acres of just corn, acres of just soybean, and so on and so on. And that has been done for decades. And there's been some debate fairly recently that, maybe monoculture systems aren't the best idea for farming practices. And if you talk to a lot of organic growers, they tend to do more polyculture, where they'll have a field where there's a mixture of crops rather than just a monoculture. And there's some thought that can be very beneficial. And so there's there's different ways to grow crops, certainly. And so now, though, this question is what can we do as we're growing crops to create a situation where the microbes, and in this case, the soil microbes, the soil microbes are primed and ready to defend the plant? Is there a way to grow those crops that can create that situation? So that's part of what she's asking here for this idea. And so we were zooming in. These are the microbes we zoomed in on are the Streptomyces bacteria. And I still work with these bacteria today. They're very different than your typical like E. coli bacteria that you have millions of those in your stomach right now. They're a little different. They actually kind of look like a fungus because they grow with hyphal growth. They just look really petite under the microscope compared to fungi. They're much smaller than fungi, but they still have that hyphal growth. So we use those terms, hyphae, mycelium. You're looking down on a colony, on several colonies growing on an agar medium right now. And what you're seeing here is the life cycle. Well, one of the phases of the life cycle is that, all right, what's going on there? Let's try that again. I don't know why my pointer is not working. Well, that's annoying. Let me do my pen. Where is it? So here, I don't know if this is going to show up. Right there, where I circled in red, that is some aerial hyphae that's growing up off the agar. And what they're trying to show here is that it's segmenting along the whole length of the hyphae into small little compartments, and those are spores that are forming. When the bacteria go through that part of their life cycle, they start to produce antibiotics. And so they're very potent antibiotic producers. Most of the antibiotics you've taken to date have been produced by Streptomyces. You have them to thank for the antibiotics that you've been taking through your childhood and into adult. And so they're very good at antibiotic production. They make lots of different antibiotics. And so we know a little bit about how they do that. And so they're hanging out in agricultural soils. Any soil system, I don't care where you go, you're going to probably find Streptomyces. They are just everywhere. And so the question is, how can we get them to produce antibiotics that can inhibit the plant pathogens that might be plants that we're trying to grow in that field? So the way to test that is actually using agar plates, and you grow the Streptomyces bacteria in those spots, and then you spread a pathogen across that plate, and then you incubate the plate, and you look for some infection. The zone of inhibition showing the Streptomyces bacteria. No, these are just inoculated spores. And so hundreds and hundreds of these tests have been done with the Streptomyces in these agricultural fields, and we can show that there's some Streptomyces that are really good at inhibiting the pathogen, which is what we want. So it's like, how do we get that to happen more and better? And so then the question is, okay, well, when do they decide, so to speak, to produce these antibiotics, these bacteria, these Streptomyces? So you can see, for example, that there was a population here of sometimes they inhibit, sometimes they don't. Well, we want them to inhibit consistently. And so we started to ask the question, under what conditions do we do that? And so that graph on the right now is showing the idea of, okay, when you grow these bacteria, stress, and the pathogen, do they share nutrients? Do they like to eat the same? Yes or no? And so this is what you're seeing on the x-axis, the proportion of nutrients shared. So humans down for the left, they don't really have a lot in common in their diet as you progress on. Definitely very strong. And then you map that, graph that against the conditions of the streptomyces coming off the pathogen. And you can see the positive correlation with the idea that if they share more nutrients, they have more nutrients in common, the streptomyces is more inhibitory. So Linda likes to use the analogy of kind of like, you know, if you're teaching elementary school and you bring the kids back into the classroom after recess and you have, you know, 10 different tables that they can sit at, and you have different cookies at each table. And so then some students will come in and they'll rush, you know, a couple will go to the oatmeal table. I really like oatmeal. Some are like, no, I don't like oatmeal. I like sugar cookies. And so you'll get the students, the kids, kind of separating out into the tables, right? Like, I like this one. I don't care. You can have those. What I like. So that kind of analogy is down here for us to see by portion. But if you have the table and all you put out is sugar is cookies, and then let them back in for recess, now, and you limit it to, okay, I'm going to do just 5 tables, all sugar cookies. That's all you get. Well, now they're fighting over that same resource. Fewer tables. And it's just sugar. And so we now here is the idea if the competing that they both the pathogen and the beneficial biocontrol agent, they're both fighting for that nutrient, then they're gonna fight with each other. I want the cookie, you're not getting it. All right, and then it kicks on their antibiotic production. And so we started thinking about that a little bit more carefully, and the data is showing that idea. It supports the idea. If they're competing for the same amount, the same nutrients, they tend to get combative with each other, fight with each other. And in this respect, the streptomyces are really good fighters because they can kick into antibiotic production to kill off their competition so that they can get more of those nutrients. Am I making sense? Okay, so that was an interesting discovery through the research that was going on in her lab. So it seems that the nutrients that are leaking out into the soil from the plants that are growing there, that's where a lot of the nutrients are coming from, that the microbes are getting access to. It's the leaky roots coming from all the different, you know, the crops that are growing there. And so the question is, okay, what happens if we have a monoculture plant system versus more of a polyculture where we have more than one species of plant growing in these fields? How do these biocontrol agents, these streptomyces, how do they behave if they're presented with just growing with one type of plant or more? And they were very interested in looking at how do these streptomyces under these different conditions inhibit or kill off pathogens. And so all of these names that you see here, these are agar plates with different pathogens that were spread across the plate after they had taken soil samples and diluted it down quite a bit. So all of these little pinpoint colonies, those are diluted soil samples that we plated first, that to grow up a little bit. And then we came back, spread the pathogens across the surface of the plate again. So Streptomyces scabies, that's the one that causes potato scabs, Fusarium, Verticillium, Rhizotonia, all of these are fungal pathogens. The Streptomyces are bacterial. So this is just an image getting you to think about kill zones again, competition. So we're looking at which soil systems, you know, when you grow a crop, as a monoculture, or if you take soil from a soil system where the crop had a mixture of crops. What's happening here? So what they see is that okay, so on the x-axis, that's the number of different plant species that soil came from. So 1 means monoculture. 16 means 16 different plant species were in that soil system. Take soil samples from each of these 16 different plant species. And then we tested those streptomyces to say, how inhibitory are they? And you can see with the graph on the right that the mean kill zone was the greatest if the soil was taken from a monoculture field. Higher the kill zone means the more inhibitory those streptomyces were. And so it's actually showing, it's kind of supporting that cookie analogy, where if you just had one plant species, that means the number of nutrients, coming from that one plant species is not as diverse as compared to if you had, 16 different plant species all leaking out different nutrients. Then you'd have a lot more variety. But with a monoculture, it's pretty minimal choices for nutrients in the sense of what are those microbes going to eat and how are they going to compete with each other. And so here you can see that in a monoculture system, we actually created a result where the streptomyces in that monoculture soil were better competitive. The path. So, and here on the left, the portion of the data saying that there were a lot more inhibitors of species. There was many more capable of doing inhibition in a monoculture. And not only that, but they did a really good job killing the pathogen in a monoculture. And so this was not what we initially hypothesized, when we first started this research. We thought polyculture system would be better, but it's actually showing the reverse for this particular question that we're asking. How do we get these biocontrol agents to be better at inhibiting or killing the pathogen? It seems like a monoculture plant growth system was actually doing a better job at creating better killers against the pathogen. And so low diversity just means a monoculture. Monoculture in this case is the low diversity idea. High diversity is the 16 species, the polyculture idea. So it really kind of gets you thinking about if you have 16 different species in that field, that means there's different carbon sources, different organic matter composition. You would think that would be better to help support good biocontrol, but it's actually reversed. We saw with a monoculture, less diverse plant community, monoculture system, we actually had better streptomyces inhibitory profiles. And so again, it's going back to that cookie analogy, where if they're fighting for the same nutrients, the pathogen and the biocontrol agent, if there's fighting for the same nutrients and the nutrients aren't as abundant, then you're definitely going to get competition and warfare, so to speak. And in this case, it's the streptomyces turning on their antibiotic production and killing off their competitors, which is what we want, because we want those pathogens, those competitor pathogens, to be killed off. And so this diagram here, you already saw the left, and then this here on the right is going to show the upper graph, you see in the left. That means they're competing for the same nutrient versus the lower the distribution means that they are not competing for the nutrient. They're kind of on the table. I'll eat the oatmeal, you eat sugar cookies. Right? So the more resources they share, the more competitive the environment, the more inhibitory the streptomyces seem to be. which is what we were going for. And so that's the fancy word for that is niche overlap, would be when they're really strongly competing with each other, or niche differentiation if they split out from each other and they're eating their own type of cookies and they don't. So it kind of begs the question, when you're trying to not use any kind of pesticide and you want to use biocontrol as your way of killing off any plant pathogen or inhibiting disease, what's the best way to do that? And so this flow chart is trying to, at least through the studies that we did, this is the idea, perhaps something to think about when you're trying to design a field that's inhibitory to the pathogen. So here you're looking at if there's low nutrient diversity, one type of cookie and not many of it, well, then you got them fighting with each other. And the biocontrol agent can become quite successful at killing off the pathogen. But if you have lots of different nutrients and plenty of it, like if you have a polyculture plants, and all those plants exuding all kinds of different stuff, then you tend to drive the process where everyone kind of has their own little niche. They have their own separate pool of food. They don't really interact with each other. They're not fighting with each other. They're all like, yeah, let's just get along and eat our own food. And so they don't inhibit each other. Don't fight. And so that's what this is trying to show on this flow chart. And so very kind of surprising set of studies that were done. This was over a couple of years of research effort, both in the lab, in the greenhouse, and then in field settings to get at this question. And very surprising, but once you work through it and think through it, it's like, okay, yeah, that kind of makes sense. If they're competing for the same limited type of food, that they might get a little bit more aggressive toward each other. And so this idea of biocontrol sounds easy on the surface, but it's a lot more, you have to be a lot more thoughtful about how you approach this. And so this was kind of a nice, not really a cautionary tale, but just kind of like a heads up that it's a lot more complicated than we thought. We need to think a little bit more about this, because I think you might have heard there's a lot of push to do polycultures. Like that's a healthier way of growing food. And the answer to that is yes and no. It depends on your soil system, what pathogens might be there. Yes, it could be great, but it could also lead to disease pressure under the right conditions. And so this is something that is more nuanced and complicated than we initially thought, and I think most people thought. And so with the phasing out of all these different chemicals and the turn to biocontrol, I think that's a great idea to use the natural microbes to our advantage if we can, but it's not so simple and straightforward as just spraying a chemical. Like we just can't sprinkle a bunch of biocontrol agents out there and say, okay, it's good, it's going to work. It's a lot more complicated. And so you really do have to do some initial research to see what could work in your particular field with your particular crop and how you grow those crops. So it's never boring, that's for sure. When you do this kind of research, it's just surprising, often very surprising, and leads to lots of other cool questions and ideas to test out in the next research experiment. Okay. So looking at the plant microbe, the integrated plant health management, this idea that using microbes to help the plant is a great idea. And I think it can profoundly impact plant health and how plants, what kind of chemicals the plants produce, which can have an impact on our health. But it really does require some thoughtful approaches and a lot of research to figure it out. And so, once you start to look at the microbes that hang out with the plant, both the good and the bad, you really do have to know what's going on if you have disease pressure in your fields that you're trying to grow your plant in. You really need to understand who that pathogen is and how do they grow and because that certainly is going to impact how you might be able to control it. So just one example is that asking the question, does the pathogen form spores or not? So some pathogens, fungi and bacteria, can form spores that are pretty resistant because it's a way for them to go dormant, and then you can spray all the pesticide or fungicide you want, and it won't necessarily do anything because they're dormant. They're just hanging out as a spore. And so those fungicides or antibacterials won't really work. If you have a pathogen that forms endospores, good luck. Endospores are incredibly resistant. And so I think I might have told you a while ago how I found endospores lurking in my bottle of ethanol that I used to sterilize my like scalpula, what am I trying to say? Scalpels, scalpels, and tweezers, and the endospores were floating around in the ethanol, and I couldn't figure out where they were coming from. And I finally tracked it down to the ethanol. So endospores are incredibly resistant. They are, I mean, they can survive for thousands upon thousands of years in their endospore state. And so some pathogens can do that. And so again, if they're in their spore state, there's not a whole lot you can do. You have to get them to germinate somehow. And then hopefully whatever you're doing will impact the so-called vegetative cell. When we say vegetative cell in the microbial realm, we mean the active growing cell, not the one that's gone dormant into a spore. And so this picture that you're looking at is just under the microscope what you would see if you do a special spore stain where all the red cells are like the active growing vegetative cells, and then all the blue cells are spores. And so you can figure this out quite readily if you have an endospore producing bacteria just by looking at them under the microscope. It's not that hard. And so that's just one example of you need to know about the life cycle of the pathogen or the pest. And then certainly it really does depend on how much of the pathogen is present in your fields. Is it a lot or is it minimal? And certainly that's going to play a role. And sometimes there's pathogens lurking, but they don't really cause any significant damage. Even though you know they're there, they just don't quite have the numbers. Again, think about warfare. There's just not enough soldiers to do any enough damage. So this idea of getting to know your pathogen at a little bit more deep level, and then who's in your soil system or in your hydroponic system, which way you're growing. So that gets to this kind of idea. You see this in any plant pathology class where they talk about the disease triangle, and this idea that you need three things to be met. You need the host to be susceptible, so that has to be one of the factors. You have to have a pathogen present, of course, and then you have to have a favorable environment that will allow for the disease to actually bloom and take root, so to speak. And so if you have all three, then you tend to see this idea where they all converge and you end up with disease. But if you don't have all three, then you won't get disease. And so this is a very simplified way of thinking, but basically the point here is that you have to have all three in order to end up with disease with your plant. So a lot of those diseases and issues are caused by insects, but certainly the fungi, bacteria, viruses, viroids, those all fit into the microbes category. And if you think about like, okay, which of those microbes causes the most amount of disease, it's fungi. Fungi win. There's more fungal plant pathogens out there than bacterial plant pathogens or viral. So fungi definitely win on that. Herbivores, you have mammals, the fuzzy animals, but you also have the insects. And all the insects are referred to as pests You often hear that term, or they might say the insect herbivore. So there's roughly 2 million insect species out there in the world, and about 1 million attack plants. That's a lot. And so this is something that's been going on ever since insects evolved. They had to have something to eat, and plants were a good option. So here you're looking at this idea of insects co-evolved with plants over 350 million. So you're looking at some insects I love to hate in the greenhouse setting. This lower left is an aphid up close. And then you see the top two images up top. Let me see if my laser pointer is going to work. Huh. Still doesn't want to work. I don't know. I've not ever run across this issue before. The top two pictures are spider mites. You can have these, all of these that I'm going to be showing you, by the way, can show up in a greenhouse setting, in your home, out in the field. And so these are something, so these two images are spider mites. So if you ever look at your houseplant, you see this, what looks like a spider web. That's usually the first sign, because the spider mites are so small, you can miss them with the naked eye. And so you really have to look. And often whenever you look for pests, you should look on the underside of the plant leaf, not the top. They're often on the underside. But that webbing, when you see that, it's a sad thing. You're really up against a fight to save that plant when it's that bad. And then this lower right image, those are mealybugs, which look like fluffy white little cottony fluffs on your plant. And then you look a little closer and you realize they're crawling around. It's not just some fluff. And so all of these can do a lot of damage. We continually have to fight these in the greenhouse setting. If you think about a greenhouse operation where you have lots and lots of plants in a very small footprint, very easy for disease to spread. Okay, see if I can get it to advance. So when you look at insects that are feeding on plants, there's different types. There's the ones that we call phloem feeders. Those are aphids and whiteflies. They say they're they do precision herbivory because they have that very petite mouth part, the stylet, that is pretty much like a needle, where it can very precisely zoom in and pierce the phloem of the vascular tissue and start sucking away the sugar water and other nutrients in there. Then you have the cell content feeders like mites and thrips that like to break open the cells of like the leaf cells. They'll start releasing enzymes and toxins to break open the cells, and they can suck away the nutrients. And then there's the chewing insects, which just come along and just chow down on the leaf. So the whole leaf goes, down the throat, so to speak. And so those are like the caterpillars, the beetles, the grasshoppers. They cause a lot of damage because they can really chew down leaves. And so, and then don't forget, a lot of insects have the ability to have stuff, microbes, including viruses, in their saliva, so that when they are chewing or piercing any of the tissue, they can introduce microbes from their body into the flesh. And so that's another issue that you worry about. And so that picture up top, those are white flies, literally white flies. Those I battle a lot with when I do my big potato experiments in the greenhouse. I remember when I was a postdoc, I had 250 potato plants growing in a greenhouse. And toward the end of the growing period, I would walk in between the benches of the potatoes, and just brushing against the leaves would cause the white flies to fly up into your face, into your eyes, up your nose. You had to have your mouth shut or they come into your mouth. It was like I was in a snow globe. It was that bad of a white fly explosion in the population. Luckily, they don't bite you and they're not dangerous to eat. So I'm sure I ate quite a few over the months that I did that kind of research, but they certainly can cause a lot of damage. And then you can see the thrips there are very, very tiny, hard to spot. And so I'm just trying to give you a few examples of some common insects that you may run across, especially in greenhouse or home settings. So here's, again, a whitefly, and then some of the evidence of their damage. So depending on how they feed, that you need to know that so that you can figure out how to try to control them. And so, you do have to know a little bit of entomology, knowing a little bit about insects in order to successfully grow plants. And so, with the pesticide application, knowing what type of mode of eating they do makes a big difference. And then last but not least, fungus gnats. You might have seen those on some of your house plants. So they're not usually an issue, but they tend to flourish if you overwater your plants, especially in the winter. So when winter hits around here, even though your plants are indoors, and yes, you still have to water them, but you shouldn't water them as much as you think in general, because they're not as active. They've kind of slowed down. And so don't be too generous with watering. And then these fungus gnats lay their eggs, and then the larvae that hatch from that will feed on any organic matter or fungi that are in the soil. But if they run out of that kind of food, then they start attacking the roots and the stems, and then they can spread disease as well as they're chewing on the plant because of what they have in their bodies. So Pythium diseases, so Pythium, Phytophthora, and Fusarium are very famous fungal pathogens of their genus names. And so they can get spread by the fungus gnat. And so as they fly around, though, they really don't do a whole lot except be a little annoying to have those in your house. But it's the larvae that can cause the most damage. So the only time they can really start to cause issues is if the flying phase of their life, if they start to get really high populations, or even the larvae. So generally, it's not a huge deal, but an easy way to get around this is to not water your plants as much. And doesn't want to advance. So again, looking at all the things that can go wrong when you're trying to grow healthy plants. On the right side, you see a lot of what we've been thinking about, with the pathogens attacking the plant, the herbivores, the insects. On the left is a lot of abiotic issues. So when you're growing plants, especially outdoors, a lot of this will come into play that you have to worry about. So the abiotic, drought, flooding, and all kinds of other things to worry about. And then I put off to the right there, I typed in weeds, because that's another issue to think about when you look at biotic interactions, is plants that you don't want in your field taking up resources and space, so weeds. And so you have to also think about them as well. Okay, so what I'd like to get into next time is looking at, as the plant is trying to stay healthy and grow and finish its life cycle, if it is getting attacked, what they tend to do is produce these secondary metabolites in response to attack. And it's their way of defending themselves. So I wanted to talk a little bit about that and just kind of give you a general idea of what they're doing. Because what they're doing when we eat those plants that have these secondary metabolites, it's a good thing for us. It actually benefits us, which I'll talk to a little bit more about. So I'll save that. It's too big of a topic to really get into when we only have a minute left. So we'll stop there. By the way, with the exam, if you want to go over any of that, maybe on Wednesday I can talk to you a little bit about that, the essay portion in particular. But if you want to go over any portion of that exam in detail, please come and make an appointment. We can do that. Okay. But on Wednesday, I'll try to cover a little bit about that in class.